Firstly, welcome everyone. This is welcome to the BBI International Webinar Series for 2021. For those of you that maybe haven't been here before, these take place approximately every second Wednesday of each month, and we've got a really great lineup of international speakers in all sorts of different areas of biodesign and synthetic biology this year. And today I'm joined by Professor Woolfson, who will be helping me field some of these questions to Elisa. So without further ado, it's a real pleasure to have Elisa Franco here today. Elisa is an associate professor in mechanical and aerospace engineering and bioengineering at UCLA. Her background is a PhD in control and dynamical systems theory, which was from Caltech, but she also has a PhD in automation and power systems engineering, I think that's right, from the University of Trieste in Italy. She's received numerous awards such as the NSF Career Award, Hellman Fellowship, and the Rose Hill Innovator Award. And her research is kind of focused around uses of kind of biological feedback within DNA and RNA nanotechnology. And she has a real focus on kind of a more rigorous design, modeling, and synthesis of circuits, and also materials, which I think we'll hear about today, using nucleic acids and proteins. I think this fits in really well with some of the kind of more molecular bottom-up approaches that we have here in Bristol. And so I'm really looking forward to see what she has to talk about today, which will be around the idea of programming dynamic behaviors in molecular systems and materials. And I'll hand over to you.
All right, thank you for the invitation and for the introduction. So let me share my screen. Hopefully everything will go smoothly. Okay, so you can all see my screen, everything okay?
Yeah, that's perfect.
Awesome. Okay, all right, so I usually start my talks with this slide that gives you a very high level overview of our motivation. Our main question is whether we can figure out ways to embed the dynamic behaviors you see in cells in a synthetic material. So the prime example is this very famous video of the neutrophil chasing a bacterium. So you can see that cell as essentially as a smart blob, okay, that is chasing that black dot. It's sensing the chemical trail it leaves behind. That blob is actually rearranging its shape, and it's moving to chase that little dot, and eventually it catches it and it degrades it. And the way this blob is working, this neutrophil is actually doing its job, is by having like an internal reorganization. Okay, there's like cytoskeletal filaments that are self-assembling constantly and self-assembling in the right directions so that it can perform its motion and its goal of catching the bacterium. So we all know how the cell is organized, right? How does it do this? There's coordination between sensors, receptors that are at the surface. Then there's signaling circuits that convert that signal that is sensed into regulatory signal that the cell can understand. And then those signaling networks tell the what DNA to transcribe and translate, what proteins to produce, and also how to regulate those proteins. It's not just about what material you're making, what self-assembly you're producing, but it's also how you're regulated. So basically that figure to the right, that kind of summary cartoon of how the cell works, if you were to think about putting it into a material, right, it would mean that you need to endow your material with sensors, circuits to do the control part, and the actuation. Okay, so you want to sense the environment, control both the production and the regulation of the materials you care about regulating. And now, so our goal is to basically do this very high level goal. And the way that we go about doing it is by using nucleic acids. And the reason why we use nucleic acids to build each of these parts and each of these materials is because essentially we have a very large toolkit of components that are ready, that are out there, and they can be grabbed and composed. So sensors, you find them in the form of aptamers, so nucleic acid molecules that switch their conformation in response to the presence of a ligand that they bind to. You have circuits. There's been many demonstrations, both in vitro and in vivo, of DNA or RNA signaling cascades and even dynamic circuits. And finally, the assembly part, which is probably one of the most striking and unconventional applications of DNA nanotechnology. You can design nucleic acids, both DNA and RNA, so that multiple strands interact in prescribed ways, and you can build really literally shapes, objects with sizes that vary between a few nanometers to tens of microns, and you can interface those objects with non-biological ligands. So they can serve as sophisticated biomolecular scaffolds for non-biological items. So these materials that we're aiming to build could really be multifunctional. Now, so there's so many components out there. Our job, our lab, tries to figure out what are the driving principles to interconnect them together. So we are really asking systems engineering level questions. How do we grab a particular sensor, connect it to a particular type of assembly? And I'm telling you, there's lots of components out there, but what is the intrinsic advantage of nucleic acids? Well, the intrinsic advantage is that you can do rational design of their interactions, which I hinted at when I was mentioning the nanostructures self-assembly. So we all know that DNA is in a double helical form typically, but when you buy DNA, you buy it in a single-stranded form, so you buy a backbone with a bunch of bases. And how those bases will interact depends on the sequences of bases of the strands. RNA is basically a more complex version of DNA. The chemistry is slightly different, but on an abstract level, we can think about it in a similar way. We can program how it behaves and the interactions of multiple RNA molecules by programming the sequence, okay? And so the basic principle is that of self-assembly. If you have two nucleic acid molecules with particular sequences, the likelihood that they will form a complex and also the speed at which that complex will form depends on the sequence itself. Because this is a process that, yes, it is sequence driven, but there is a multitude of sequences that can satisfy the same requirement of binding and the same requirement of kinetics, we can stop thinking about sequence, but the specific sequence we choose, and simply refer to them as domains, okay? So in the rest of my talk, I will not really show you specific sequences of my oligos because I don't care. They could be anything as long as they satisfy the complementarity requirements I want. And so finally, of course, we have good methods to do this kind of mapping. From sequence to folding, we can go both ways. We can start from a sequence that is given to us and predict how it will fold, either alone or with other oligos, and we can go the other way around. Once you have a target folded structure, there are ways to optimize, to figure out what's the optimal sequence to give you that particular folding. And of course, it helps the DNA is custom synthesizable, and it's very cheap. And an advantage of RNA is that it can be encoded in DNA and potentially expressible in cells. So these are very interesting materials to work with. All right, so this is basically the high-level spiel of why we do what we do. Now let's go into the detail of what I'm going to present to you today. So I'm going to start by telling you about a particular class of assemblies. So here I showed you a picture where there's like many different kinds, from few nanometers to like tens of microns. You can make even crystals out of DNA that are 100 microns in size. But we wanted to, because our interest is in dynamic materials, okay, materials that like cytoskeletal filaments can assemble and disassemble. Basically, we wanted to focus on a particular class of assemblies where you can use nucleic acid inputs to determine how that assembly behaves. And we really focus on filaments that are basically an artificial version of actin and microtubules. Why? Well, if you think about it, they are very simple architectures, very simple assemblies. You have a few types of monomers that by controlling how they react between each other and with some organizing proteins, you achieve a very broad variety of assemblies. Okay, so you start from a filament, but by changing, how the monomers nucleate, how fast they polymerize and depolymerize. You have a few organizers that either cap or interconnect the filaments, and you can get, filaments of different sizes, bundles, networks, meshes, and that's how the cell accomplishes all its spatial organization. And the key idea is that these monomers, they assemble at room temperature. It's nanometer elements that form micrometer structures. And it's also a reversible process because the material gets recycled all the time. And cells use the filaments for a variety of things, right? For driving complex processes like cell division, to move, and to transport stuff. So the equivalent of this in DNA nanotechnology is DNA filaments that we call tiles. And let me tell you, there's many variants of DNA filaments. And I'm going to focus on one particular class because it's very well studied. And it allows us to achieve very fine control over how they assemble and disassemble. So these tiles, as you can see them here in my slide, they are formed by 5 distinct strands. Each strand here is represented in a different color. So we have yellow, blue, and green. And these strands, they cross over from the top helix to the bottom helix through two crossover junctions. The red and the purple, they don't. The red and the purple, they just run through their own slide without helix without crossing over. So as you can see, because there's 2 crossovers, you can probably get a sense that this is a rigid monomer. And it has some domains that are not paired, they're single-stranded, and I'm calling them A and A prime. These 2 are complementary to each other, and then B and B prime. So if you design this tile right, it will form lattices, and the lattices have a curvature that will, they will end up closing on themselves and forming hollow nanotubes with an internal diameter between 7 and 20 nanometers. It really depends on how many tiles start the nucleation process, and they basically close this tube. And so I want to also tell you that those single-stranded domains, we call them sticky ends, and they're like connectors in Lego blocks. So here I'm showing you an example image, fluorescence microscopy image of these nanotubes. As you can see, they get pretty long, although the individual tiles are only nanometer, 14, 15 nanometers. How do we see them? Well, we place fluorophores in, basically an arbitrary position on the tile. Usually we put it on the blue tile. I'm not showing that fluorophore here, but it's how we see them. And the idea is that if you self-assemble the monomers, so if you form this double crossover tile and you mix them, then they will form polymers that elongate for a really long time. And I'm going to show you a lot of plots where I show you time in the x-axis and mean length on the y-axis. So basically this shows you the mean length over time with error bars that are derived either by multiple repeats of experiments or by bootstrapping. So as a side note, you can make nanotubes, similar nanotubes also with RNA. We have shown that in a series of papers. These are a bit more tricky to obtain. You need to adapt the geometry of the tile to A form, to helices in A form. And the RNA is more sticky, so we had trouble growing these nanotubes, but we can form them, and they have similar length. But this is kind of like a side note. So now the key that what we wanted to do was to figure out ways to control dynamically how the tiles assemble and disassemble. So basically it's about understanding how to activate and deactivate. So when the tile is active, you get nanotubes. When the tile is inactive, you should get disassembly. And we studied different ways to do that so that we mimic what happens in nature at a high level with microtubules. So you have nanometers elements that give you micrometer structures in a reversible process, and it all happens at room temperature. So I'm going to now show you, just to give you a broad overview of like work we have done in this space. So we were able to control self-assembly of these monomers, of these styles, using pH by using a DNA sensor for pH, something that changes structure depending on the pH level, and then that signal is transmitted to the tiles, and by simply changing pH, you control assembly and disassembly of the nanotubes. Now, that would normally, they would not respond to pH, of course, they would be completely insensitive to that. We have figured out ways to direct assembly through RNA. So we have designed tiles that are inactive constitutively, but as you have transcription of an RNA molecule, they get activated and you have assembly. And then you have enzymes that can degrade RNA once it's incorporated into the tiles or in the structures and causes disassembly to happen. So this is how we obtained transient assembly with a programmable duration of the assembly. And finally, we have also developed a system where we interconnected a molecular oscillator. This is an in vitro oscillator formed by that is transcription only. There's no protein translation. And this oscillator has been characterized in a series of papers prior to this work. Now you can use some of the outputs of the oscillator to direct assembly and disassembly of the tiles. And this was an extremely challenging project. This would be a whole talk just by itself to describe how we did this and what are the challenges we encountered. But at a very high level, the problem is when you design reactions that are even indirectly coupled and they are out of equilibrium, where the, with behaviors like oscillations that are strictly parameter dependent, it becomes very difficult to tune everything to operate in the correct regime. So we were able here to demonstrate at most 2 oscillations. So you can see there is a lot of room for improvement. But in these 2 oscillations, we were able to get nanotubes to break. So here at the peak in point C, once this invading species, this thing that breaks the nanotubes is at a high level, the nanotubes break. And once at point E, that species is low in concentration, the nanotubes reassemble. And then over time, their concentration goes down again when the invader goes up. So we were able to demonstrate 2 cycles. Now, what I want to talk about today more in detail is instead a more recent project where all this toolkit of methods to control assembly and disassembly of this biological material, we explored how it would work if we were to encapsulate it in a mimic of a synthetic cell. And so I will talk a little bit about, how we did the encapsulation into the compartments, how we developed the data processing, and how we achieved RNA-directed assembly in a transient way. And this is a project that was driven by Melissa Klock, who recently graduated, and Sid Agarwal, who, Sid is still a student in my group. And this is a manuscript submitted. I'm happy to share if you're interested in reading it. So first of all, what does it mean when I say a synthetic cell, a small compartment? I'm talking about water in oil droplets, which is probably the simplest way to encapsulate biological or chemical components. And there's different ways to create emulsion droplets. One is by shaking, vortexing 2 phases, aqueous and oil. And the other is through microfluidics. Those are the 2 probably simplest ways, non-chemical ways, let's say. And so in one case, the emulsion the vortexing method doesn't really give you much control over the size of the droplets you get, whereas microfluidics allows you to do that. But one major advantage of simple vortexing of the sample is that it's very quick protocol, and you can get lots of data with on droplets that have diverse dimensions. So everything I'm going to show you today is data generated by simply taking an aqueous phase containing the DNA and then an oil phase, vortexing the sample, and then observing it under a microscope. So we demonstrated that you can achieve an encapsulation of nanotubes in different ways. So for example, you can take the 5 constitutive strands of the tiles that I showed you, anneal the tiles and form the nanotubes, and after that you can encapsulate. But as you can see, the quality of the nanotubes you see in these droplets is not super, because they tend to probably, like the encapsulation post-formation essentially creates defects and creates stress on the crystalline structure of the nanotubes. So they are still there, but they tend to form these bundles and like hairball structures that we don't particularly feel is controllable. So we're not, we did not use this protocol much further, but you can do that. Another strategy is to first encapsulate the DNA strands and later anneal once they are encapsulated inside the droplets. And that is something we can do because these droplets are very resilient to heating. And in that case, as you can see, we get more crisp, well-defined structures. Now, the problem is that, of course, they start forming throughout the annealing process, and they may be difficult to observe as they develop. We can show that you can, because so I told you at the beginning, the way that this particular sequence I have in my DNA tiles doesn't matter. So you can actually build distinct populations of nanotubes by simply building distinct tiles that have distinct sequences and don't interact with each other. So in this case, we can have like a red tile and a green tile, and you can encapsulate them all together, and then you anneal, and you will form distinct populations of nanotubes that don't necessarily interact with each other. So here you can see the merged channel of tiles of nanotubes that are functionalized with different fluorophores, and you can see them in different color. But what the system that gave us the biggest ability to control assembly is one where we have two distinct tiles, okay, and only in the presence of both you get assembly. So only when both the red and the gray tile are in your solution, you get nanotubes to form because of the way the sticky ends are designed. So basically, you can pre-anneal, pre-form the monomers, store them in separate vials, and then only once you mix them and you vortex, then you can obtain nanotube formation inside the droplets. So here is an example of how we of nanotubes growing inside droplets over time. These are 3 droplets that were tracked over the course of 24 hours, and you can see how initially you see the droplet has a uniform fluorescence because you only have tiles that are not assembled. And as time progresses, you can see how filamentous structures start to appear. And at the same time, the background goes away because the tiles are being incorporated into the nanotubes. And by the end of the experiment, around 24 hours, the nanotubes have actually mostly condensed into few very long structures that could be individual nanotubes, but could also be bundles of nanotubes. And they seem to be quite rigid, and they seem to form rings around the diameter of the droplet. So I'm going to now show you a movie of that process here in the same slide. So as you can see, over time, they wiggle, they start condensing, they start assembling, and then by the end of the experiment, they really look like long, rigid sticks inside the droplet. So this formation of rings is actually, let me see if I can play this movie. Yeah, so here you can see this is a replay of like the last few hours of the experiment, of the images that I showed you earlier. So this phenomenon of forming rings is observed also for other types of biological polymers. So there is lots of both experimental and modeling work that shows how this is, kind of a universal behavior of biological polymers. And so you see images of microtubules inside droplets that form these ring-like structures, and also actin in vesicles. And it really depends on properties of the polymers that include their persistence length, their diameter, and of course, how they interact with each other. So this is something that we have not studied in depth yet, But it's I feel it's a very interesting, cool behavior. Anyway, our goal for this project was figuring out how to actually track multiple nanotubes in multiple droplets. And turns out that that's actually very hard. So this is a confocal microscopy image that we got after a lot of labor. So as you can see, it shows you a lot of specks that are really concentrated near the surface. Okay, so those, I told you there were rings. Yes, those are rings. The nanotubes tend to align in the internal surface of the droplet. But then how would you, from these images, it's really difficult to get a sense of how many you have, how long they are, and then that's because they intersect, they overlap, and they move. So if we wanted to get some sort of more objective quantitative measure of assembling in droplets, we had to figure out a simpler method than trying to get their length and their number. That's not going to happen. It's very difficult. So what we did is that we used the statistical properties of images. So you know how I observed before that initially the droplets have uniform fluorescence, okay? And here at the bottom is for this particular droplet, you have a plot of the number of pixels versus their brightness. And as time progresses, this uniform fluorescence changes. And now you have like darker spots and then brighter spots that correspond to the nanotubes. And as you can see, the shape of the histogram of pixel intensity has changed. And this trend continues over time. At the end of the experiment, the pixel histogram, the intensity, as you can see, it looks like it has a peak and then a really long tail. And it's quite different than what we had at the beginning. So we figured it would be a simple thing to just look at statistical properties of the droplet images to figure out what's the extent of condensation. And to extract the change of shape from the histogram, we used two measures that are the third and the fourth moment of the histogram. So skewness, which gives you, quantifies the symmetry of the histogram. So skewness is positive if you have a peak at the left, basically, and a tail to the right. It's negative if the peak moves to the right. So skewness tells you about symmetry. Kurtosis is the fourth moment, and it tells you about the tailedness. And to figure out whether we have successful formation of some assembly, we want to look at both. It's not enough that we have an asymmetric histogram, but it also should have big tails, especially on the right, because it tells you have a number of very bright pixels. And so here is an example, like if you were to take this particular droplet and track the histogram of the pixel intensity and measure skewness and kurtosis, this is what they would look like. They start near zero, and then over time they increase. And so this is what we decided to do for experiments where we have hundreds of droplets that where we want to inspect what's the level of assembly. And I should say that the shape that the histogram has it's sort of independent of the mean. So this method is insensitive to bleaching and exposure changes as long as you remain in the linear range of the detector. So in that sense, it's a quite robust method to qualitatively measure assembly. So here is an example where we monitored assembly of our nanotubes at a particular concentration of tiles. And so you can see here that both skewness and kurtosis in orange, skewness is in purple, kurtosis is in orange. They both go up over time. Now let's see how this could be useful. Well, what if we want to compare how this experiment progresses relative to a different experimental condition? For example, if you have everything left and nothing changes except we add PEG, which is a crowding agent. So PEG, because it essentially excludes volume, it forces assembly to progress faster. So if we compare images, we can clearly see that nanotubes assemble sooner But if we want to look at across like a variety of droplets and just do statistical analysis, what we can do is we can use skewness and kurtosis. And here you would see that you would achieve a plot that differs from the top one in the initial values of skewness and kurtosis. So the difference starts, you start noticing the difference from the beginning. And it's you know, in this case, this it is a sound comparison because they essentially start from the same background. You have the same amount of tiles, and because they measure the change in the histogram shape, you can starting from the same background, it basically allows you to get comparable results. Now, if you were to start, like there's a problem with this method, however, that we found out. If you actually start from different backgrounds, then it's difficult to compare the results. So here is an example where we have, you know, in one case, a low concentration of tiles, and in the other case, a high concentration of tiles. Now, at the bottom, you can see the nanotubes start forming sooner, but that is not really reflected in the plot. So the initial assemblies actually looks slower here in the bottom plot, and that is because you have a higher background. So you're always comparing the bright areas to the dark areas, and because of this, the background is different. Essentially, you can't compare. So this is a method that is useful, but it has to you have to use some caution when you apply it. All right, so now let's, how am I doing with time? Can I go on for another 5 minutes? Is that okay?
Yeah, that's absolutely fine.
Okay, all right. I mentioned earlier, we developed a method to control assembly and disassembly using RNA. So when you have RNA production, the tiles become active. And then if you do have enzymes that degrade RNA, they deactivate the tiles. And how does this activation work? Well, we simply have a DNA tile that is modified to include one RNA molecule shown here in green. And let me remind you again that these are completely arbitrary strands, completely arbitrary sequences. So the way we design them is that we simply enforce complementarity domains, and we minimize secondary structure of individual strands, and we make sure that the complementarity follows this pattern. So, and the way you do it is, for example, using NUPAC. Okay, so now we showed that this reaction, that first we characterized it in bulk, can happen inside droplets. And here is you know, an example of what you would obtain if you were to encapsulate RNA that has been extracted together with the tiles inside nanotube, inside droplets. Now, one thing that we observed, however, is that if you have too much RNA, the assemblies kind of get they're not as well formed. And in fact, here you can see they basically reduce to aggregates. They're no nanotube any longer. And we think that because excess RNA is a lot more sticky than DNA, it may actually bridge different tiles without allowing them to form nanotubes. So this shows you another pitfall of the statistical analysis method that I mentioned, that we developed, in the sense that it's not able to distinguish whether you have the correct assembly or not, that you can only inspect by eye. So, however, if you keep the concentration of RNA low and close to the stoichiometry of the tiles, so close to 1x, you do get filamentous assemblies. And so you can essentially, as long as you keep that in mind, you can proceed with this type of reaction encapsulated. So the next step was to have monomer production and transcriptional programs inside the droplets, okay? This was to obtain temporal control of assembly that is driven by, say, a genetic circuit, a simple artificial genetic circuit. And so these are non-encapsulated experiments where we only varied the concentration of gene. And of course, here we call it template because it's not really a gene. It's just a short linear sequence of DNA that transcribes an RNA species. So as that species increases, you get faster assembly, faster formation of nanotubes. Now, if you do this inside droplets, you obtain a similar behavior. So this is an example image of how initially you have no nanotubes, but as transcription kicks in, they form. And if you compare in droplets how quickly this happens, you see an interesting behavior, which is at a certain concentration of gene, the skewness, I'm showing you only the skewness, but we obviously tracked also kurtosis. It goes up over time and then it plateaus, meaning that you achieve assembly of most of your tiles. As you increase the amount of gene, that plot goes up faster. Of course, they assemble sooner. And as you increase the gene concentration even further, it basically goes immediately to a plateau, but also this curve looks like it shifts down. And that is probably because of what I was mentioning before. Too much RNA causes the formation of aggregates. And that means maybe not all tiles are getting incorporated into the assemblies because they're very irregular and full of defects. But we have not fully characterized this phenomena yet. Now, what if you had to include both production and degradation? Well, in that case, there's lots of different behaviors you could achieve. What we saw in non-encapsulated experiments is that you get transient assembly. So it's having both production of RNA and degradation generates a pulse of nanotube assembly. This is, our understanding is because the production rates are faster initially. So we transcribe RNA using T7, and T7 is known to have a burst of activity initially. And also, depending on which brand of polymerase you use, they have a different lifetime. So the particular combination of enzymes we chose is such that the lifetime of polymerase is shorter, much shorter than the lifetime of RNAs. So initially, production dominates, and you have formation of the nanotube, so the mean length goes up. But as RNA polymerase loses activity, then RNase H takes over, and basically the nanotubes go away. Degradation dominates toward the end of the experiment. There's other phenomena here, but I won't go into the detail of that. Now, the idea is that we wanted to see if this could also be obtained inside droplets. And in this case, we would see inside our small compartment that nanotubes would form only transiently and then go away. And in fact, this is what we observed. So the bulk reaction that we characterized is portable to this little synthetic cell where you can run essentially the same thing. And we also showed that you can tune, as in the bulk reaction, you can tune the pulse. You can tune, how quickly the nanotubes form, and also how quickly they go away, and what is the mean length of the nanotubes. In this case, by tuning RNase H, but there's other parameters you could change. And so this is, in a way, a programmable behavior inside the compartment. So with that, I am almost done. I kind of gave you an overview of this recent project that aims at taking DNA material assembly and port it into a mimic of a synthetic cell. Now, what's next? So I talked about at the beginning how the cytoskeleton takes advantage of filaments, but there's also a lot of other organizing proteins that hold everything together. So here on the right, there is a very pretty picture of the formation of the mitotic spindle, where you can see colored in different proteins in different colors and then the chromosomes that tell you how the microtubules, like all the whole cytoskeleton, forces a cell, polarizes the cell, and then splits into the genetic material. So the key here is, well, there's multiple components that are working together, and what if we could try to do something so sophisticated using DNA nanostructures? And so that's kind of like a grand goal that we have next. We want to take multiple components, multiple types of DNA nanostructures that could organize the nanotubes and place them inside our artificial cells. In particular, we will use origami. Here is an example of what they are. So the initial thing we're aiming at is to take DNA structures that work as struts, that they can be L-shaped, T-shapes, or Y-shaped, and then have them grow in different shapes inside droplets. And this is a project in collaboration with the Schulman group at Johns Hopkins. And another thing that we're currently working on is to introduce communication between droplets, so you could develop some sort of differentiable scaffolds in different droplets, depending on how they communicate, like what signaling molecules are propagated in the population. And this is a project with Fritz Simmel. So with that, I will conclude. So I hope I gave you an overview of the motivation of the research we do in my lab, with particular focus on the creation of responsive DNA scaffolded materials, and how do we embed dynamics in them, in particular if we work in small compartments. And then so finally, I want to thank all my group. There's here it's a bit of an outdated photo, but it gives you a good picture of like the people who contributed to most of these groups. And of course, I need to thank the funding agencies that made it possible for us to pursue this research. And with that, I will take any questions.
Well, thank you very much, Elisa. That was a really beautiful talk, I have to say.
Thank you.
Beck, are you here? Yeah,
I'm here. Yeah, that was lovely. Thank you very much. And we've got a whole bunch of questions here.
Okay.
An unlucky 13 at the moment, but I'm sure there'll be more in a minute. And a couple of those have been upvoted. So the top of the list is from Steve Mann. If you use a surfactant to stabilize the emulsion, how does changing the surfactant head group influence nucleation and growth of the DNA nanofilaments?
Yeah, of course it does influence. It changes everything. So let me actually go back to one slide where I am giving you more technical information about the surfactant, because I went fast on that. Okay, so the hold on. Here it is. I have an extra slide. So we use a particular combination of fluorinated oil and surfactant. So the surfactant is a rather big molecule that it has been used in a series of studies with encapsulation of DNA molecules, and it is based on what we found, it's the best one. It minimizes interactions with the surface, and it also doesn't seem to be charged and attract DNA. Now, that said, there are still some weird effects we have noticed that depend on fluorophore, not really on DNA. So DNA seems to be working really well with this particular surfactant oil combination, but then not all fluorophores behave the same, depending on their hydrophobicity. Some fluorophores actually make it so that the structure tend to adhere to the surface a bit more, and that also affects how they assemble. Both nucleation and then polymerization, it tends to happen at the surface. We tried other types of surfactant, but they didn't work as well. But that's a question for synthetic chemists. It's not, we kind of like grab what is available to us. We're not into the development of better surfactants.
Thank you.
I hope that answers the question.
Hopefully that answers Steve's question. Another one's been massively upvoted now. So top of the poll now is, do you see the filaments pushing on the walls of the droplets at all? And I guess if not, could you do this and mimic cell remodeling by the cytoskeleton?
That's another thing that we really hoped to do, like using these nanotubes to then induce changes in the shape of the droplets. Now with these droplets, that's probably not going to happen, precisely because these surfactants are rather big, and the surface tension is just not right. Like the nanotubes cannot deform them, basically. If you do a back of the envelope calculation, that can't happen. We tried to encapsulate in vesicles, so like lipid bilayer, like liposomes, where we were hoping we could tune the rigidity. Now that is actually very challenging. It's challenging because, first of all, it's challenging in general to obtain liposomes in the size range that we care about. So between 5 micron and like 50 micron diameter, it's very hard to get vesicles in that range. You can get micelles really easily and giant liposomes very easily, but that intermediate range is hard. But not only that, the methods to make vesicles are generally like harsh for DNA nanostructures. So we have not yet figured out a viable protocol to do that. We think microfluidics are probably the answer. But at the moment, there's very few groups in the world that can produce liposomes in the size range that we hope to get, and with microfluidic approach. So there's a lot of work. I mean, I think there's like papers in the making that will come out in the next months. People who are trying to do this, but we weren't able to yet. Although I agree, that's really exciting. That's what we were wanting to do.
Tom, how do you want to handle those questions? Should we ping pong, or shall I just carry on?
You can ping pong. So I've got one that's related, I think, to like functionalization as well, which is like, do you think you'd be able to recruit DNA binding proteins or other things to your nanostructures by inserting certain recognition sequences?
Well, I think so. We haven't tried, but I think that that's possible, absolutely. In fact, one funny thing we discovered is that T7 polymerase, if you don't design the nanotubes right, it likes to bind to them and transcribe mostly crap from the nanotubes. And now we actually published a paper on it because it's something that we couldn't explain. And it was a really vicious mechanism. So the T7 would bind to the nanotubes, transcribe RNA, that then was, of course, a copy of the DNA in the nanotube. And then that RNA would come back and disassemble the nanotubes because it interacts with the sticky ends. So it's really something we did not expect. And but yeah, I'm sure that if you add in there some recognition domain for particular proteins, they will bind.
What would you just on that? What was the ribonuclease you bunged in to break these things apart? Because
RNase H. Sorry,
it was H. So that deals with hybrids as well.
Yeah, that's yeah. We that's precisely why we want it, because we wanted to degrade only RNA that is already bound to DNA. Yeah.
Okay, so if it's ping-ponging, it's ping-ponging back to me. Is that right, Tom?
That's right, go for it.
So the next one down is, can you branch the filaments by design?
Yes, you can, using these organizers that my colleague at Johns Hopkins makes. So you can, let me zoom in into here. So these red, I don't know if you can see, sorry, it's Zoom. So these red tubes here, these are also made with DNA, and they're made with a very different method than the one we use here. It's called DNA origami, and you get much more precise control over the nano scale geometry in that case. So they don't grow as big, but you can control very well their shape. And so as you can see, they can be L, T-shaped, Y-shaped, and I bet they're working on like different shapes as well. And that would allow you to get nanotubes to branch in particular spots.
Cool. I've got a question about I think the statistical analysis. So do you mask the background when considering working out the statistical?
Yeah. So the algorithm is actually not that simple to do it. My student Melissa, she worked out a method where, you first look at the images, and you need to generate a mask for the individual droplets. So in that case, you play with the background a lot, because you need to get a clear, sharp image of the individual droplets. And then after that, however, as I said, it shouldn't, as long as you're in the linear range of the instrument, you shouldn't get different, like the results don't depend really on exposure or on like background filtration, as long as you're in the linear range. But we do have to do a bit of masking and like changing background just to identify the droplets. Otherwise, it's really hard, because like, as you can see toward the end, especially toward the end of the experiment, when you have bleaching, sorry, let me show you another slide. The edge is kind of difficult to see, right, over here compared to the beginning. If you look at 0 minutes, you can clearly see the spherical droplets with like fluorescent material, but toward the end, it's hard to detect the droplets.
Back to me, Tom.
Yep.
So this is a clever one. Somebody's used the name bit of the slide now to enter a second subsidiary question. So I'm going to ask 2 questions in this one. How do you think these materials can be used in the real world, in real world applications?
That's a good one. I am not 100% sure. Like, we envision using them within synthetic cells, and like for very fundamental studies of, you know, conversion between chemical and mechanical energy. That's the high-end goal, which we haven't gotten there yet. Because DNA gives you a very good toolbox to control lots of parameters, to control the geometry, to control the kinetics. So we really use it as a like basic toolbox of showing what you could do if you could control all these things, if you could control kinetics, if you could control how things interact with other chemistries. Well, this is how you would do it. Does that make sense? I don't see at the moment, like aside from synthetic cells, at the moment I don't have other particular applications in mind.
So I guess it comes on to the subset question from this person. So what are the properties that you expect for these DNA-based materials to have compared with commonly used polymers and materials?
Yeah, so, well, the main advantage is that, again, you can interface them with variety of reactions. Okay, so you can build both the chemical reaction and the DNA and the assembly with the same type of substrate. So it's easy in a way to obtain a dynamic behavior because you can program both things and they're both compatible. Like, you can generate a DNA-based circuit, will generate some output, say, you know, a pulse, and then that pulse is immediately processable by the assembly because they use the same chemical. So that's one big advantage. Another advantage is the fact that at the structure level, but also the circuit level, they're easy to interface with non-DNA chemistry. So, as I mentioned, you have aptamers where you can take, I don't know, ATP or you know, metals, and you can sense a variety of ligands and then communicate that event to the structure using this aptamer translator sensor. And same, so also at the level of the structure, you have the same advantage because you can decorate them with a variety of ligands. You can decorate them with metal nanoparticles, you can decorate them with proteins. And so they are very versatile, both chemical communication components and scaffolding components. So that's why I see they are helpful in general for material science. Now, of course, they have a lot of other like drawbacks that I have not mentioned. For example, you know, the DNA degrades, you know, they're sensitive to temperature, but I would say that that's also true for other chemistries. So perhaps it's not too bad as a disadvantage, but, you know, from the perspective of interfacing with the non-DNA world, they're pretty good and give you, they're kind of like a breadboard for programming behaviors.
Yeah,
that's cool.
Thank you. Tom, over to you again.
Yeah, so I've got one about using these things, I think, in living cells. So when using them in vivo, how does molecular crowding affect function and assembly dynamics? But I guess also you've got active processes in the cell as well. So would they potentially have effects in terms of how these things assemble and work?
Yeah, so I think that that's the next frontier of DNA nanotechnology, is to figure out how to operate both circuits and structures inside cells. And there's evidence that there are chemical modifications, for example, phosphorothioation of the backbone. You're replacing essentially the phosphate with a sulfur molecule, and you can protect them from degradation. That is obviously, like, the cells degrade single-stranded DNA, so it couldn't survive. But if you protect the backbone, then that process is significantly slowed down. Anyway, so there's a lot of work in that direction. Now, in terms of crowding, it's unclear how that would affect assembly, because it could, depending, you know, on where the production is localized, it could actually enhance it. If you create local, more dense areas, but it could also slow it down because it would limit diffusion. So I don't, you know, we have not done experiments yet in that sense. I don't have a sense of how it would play out. But I will tell you one other thing that we are interested in right now is in actually producing RNA assemblies inside cells that would phase separate, that they would form aggregates that don't necessarily have sophisticated control over the geometry. So, you know, like the shape of the tile that I showed you before. Let me go back to the image here. So, you know, here you have like very fine control over how these DNA molecules, they wound up on each other and where they cross over. The nanoscale control is very precise, but maybe that's not necessary for certain goals. Like if you want to simply form an aggregate using RNA, where that aggregate has a certain you know, internal motility of components, and it recruits specific components from outside the cell, then you don't need this level of rigidity and organization. So that's what we're interested in like basically generating aggregates that phase separate without the sophisticated geometry, but where you can still control, you know, when it forms, when it goes away, what it recruits, and maybe even what size, average size it has.
Tom, I have to drop out for a minute, so could you ask the next question? Is that okay?
So we've got time for one more. So this is the last one. This is always ominous which one to pick. Well, the next one on the list, which we haven't actually spoken about, is do you have design tools, or I guess software, to make knowing what sequences need to be synthesized easier? So you mentioned, I think it was NewPack, but do you have kind of more complete integrated software tools for designing some of these things as well?
So there are, but depends what you want to design. If you want to design DNA origami, there's a whole CAD software, CAD type software. It's called CAD Nano. And you can generate the DNA nanoscale object like shape and route all the strands in ways that are acceptable for DNA. So it's a really geometry based tool. And then it will also, I think it also generates the sequences for you, but I'm not 100% sure. We don't really design origamis very often. For these particular types of tiles, the tools we use are very simple. We have been using actually MATLAB scripts that were originally produced by my former research advisor, and they work pretty well. They're not fancy, okay, but they give you very control over a lot of parameters of the assembly when you do the optimization. So you can prescribe your scoring function. You can tell the software what subsequences you want to avoid, whether you want to avoid repeats, or say you don't want the T7 promoter to appear. So you can specify all those things, and it runs reasonably fast on MATLAB. But there's a lot of like tools for RNA that are coming up, both for designing structures and kinetic behaviors. I don't have a list right now, but if you're interested, please send me an email and I can forward you some information.
Cool. Thanks very much. Dec, I think it's in the interest of time, I think we probably want to wrap up, I think
I agree, but if Elisa's free, could we keep her online for a little bit for people that do want to hang around? And
yeah,
I certainly want to ask questions. So
of course, go for it.
Yeah, but yeah, should we let everyone else drop off? And
if they need to, yeah, that's absolutely fine.
I'll leave it to you to wrap up then.
Okay, yeah, I just want to thank you again for an absolutely wonderful talk, and thank everyone for attending today, and hope to see you next month.
Thank you. It was nice meeting you. Thank you.
Bye, everybody that's dropping off, and those that want to stay on. Yeah. We can talk for 5 more minutes or so before you get breakfast, I guess, Elisa.
Well, yeah, it's fine. It's 9. I'll probably just go for a run.
Yeah, oh, that's nice. So I have a self-indulgent question, which is, I used to work on protein filaments, and we're getting back into that now to try and make these things in cells and in encapsulated systems. I mean, how do you think these things compare with people that are doing self-assembly with peptides and proteins?
You mean when you say pair?
Compare.
Oh, okay. Yeah. Sorry.
But pairing would be interesting too, right? Yeah.
Yeah. How do they compare? Well, it looks like protein engineering has come really far. I have like seen like recent papers on either Nature or Science, like a very striking honeycomb assembly by like David Baker's group.
That was
So
David,
think,
yeah.
yeah. So I think that
we built one too 6 years ago, but anyway.
you know, I'm sorry if I don't know, but it's not
joking.
my field. So, but it sounds like it's protein engineering is getting close to DNA assembly in terms of generating almost arbitrary shapes. Of course, like proteins actually, I think, would be more powerful because they can also have like functions, right? As opposed to like the DNA nanostructures, once you make them, they're like very nice scaffolds, but they can't really do much aside from, say, maybe like opening, closing, or moving, but they don't have like catalytic ability. So that's, I think, where we miss out in
But I think I agree with you. I work with Andrew Turbofit a little bit. You know, the ease of, you know, going from a design concept to realization with DNA is just, or nucleic acids is just astonishing, right?
It is.
But then it's the functionalization. Yeah. So yeah, but it's fascinating. It's just fascinating what you guys can do now, and it is a lot harder. I mean, David's making it look easy, and we did it 20 years ago with small peptides, and it looked quite easy then as well. But actually getting the functionalization and the control is a difficult thing. You know, getting control of dynamics. I mean, we'd all love to make microtubules to, you know, assemble and disassemble in a dynamic way that you showed. That would be amazing. But that's, I don't know, maybe David's done it or doing it, but that would be a holy grail, I think, to do something like that.
Yeah, I agree with you, and I didn't say anything about it, but that's part of the usefulness of these scaffolds in artificial cells, potentially, because you can really adapt both the chemical reactions you can build and the shape of the connectors and the shape of the nanotubes in ways that you couldn't really do the same with microtubules or actin. They're like very difficult to re-engineer.
Oh, absolutely. I mean, and that's what we're trying with the peptides now. Can we, you know, you build something and then can you re-engineer? The nice thing about David's approach is, of course, you have a design concept, you build it on the computer, and then you build to that. So it is always difficult, as you say, to re-engineer things. But, well, I'm just rabbiting on now, and I'm conscious it's 5 past the hour.
Yeah, I had a quick question. So I was interested. There's also a lot of like interest in these kind of like not artificial chemistries, but different chemistries and what biology uses. So like XNAs and other sorts of things. Have you looked at all at using any kind of non-DNA based kind of polymers, but in the same way for working with some of these things?
Not yet. Okay. Certainly, that's interesting, because I mean, I looked at some point into PNAs and LNAs because they have much higher melting temperatures, and so you would make the structures a lot more resilient. But then when you work with those, you need to be a bit careful about how they will actually affect, especially if you look at kinetics of assembly.
Yeah.
So because we don't have good models for those, they haven't been really characterized too well. They can make unpredictable effects, but it's certainly interesting. Like my lab doesn't. We don't do that, but we know they exist.
The chemistry is also a nightmare, isn't it? I mean, you can't just order them from wherever you order them from.
Oh yeah, like not to mention they're more expensive. Yes.
Yeah. I mean, the PNA concept, I remember when it came out, I remember talking to people like Sam Gelman and thinking, wow, this could be a Nobel Prize, right? It is a brilliant idea. And I think it has realized a lot of potential, but we still don't have a real structural understanding of how they actually work. So yeah, and then there's a chemical access to them is the pain.
Yeah.
I'm going to drop off, but I wanted to say I thoroughly enjoyed that. It was absolutely fantastic. And
good, thank you. I know this is more a SynBio talks, so like my talk is a bit exotic relative to what you guys do.
Well, no, Tom will say, you know, that if you look at what we do, we just do what you do, but with proteins, right? So peptides. So no, it was brilliant for me. It was fantastic.
Okay, thank you.
So thanks for. I'm going to follow your work much more closely now.
Oh, thank you.
Doing it? Yeah, okay. So yeah, so I had some kind of more general questions I've written down here, which I think is kind of, we'll find a little bit more about you as well, sort of personally, your interest in the field and the science and that kind of stuff. So the first question I've got is kind of basically what got you started in science more broadly, and then eventually into kind of synthetic biology and the kind of artificial materials work that you're doing now.
Okay. So how did I get started in science? Well, as a kid, obviously, I was interested in animals and nature and all that. So, you know, my pipe dream was to become like Jane Goodall or something, you know. but then, so I come from a you know, working class family. So I'm the first to go to college. So you know, when I chose college, it was more about what is a degree that's going to give me the highest chance of getting a good job, because I couldn't afford, you know, staying in school for so many years and not getting a job. So I picked electrical engineering purely based on chance of finding a job afterwards. And I never really thought I could like become a college professor, just because really that's not in my family. But then a mentor took an interest in me, one of my professors in my senior year. And then I did my thesis with him, and then I got a fellowship for a PhD program. But that was my first PhD in engineering, in controls. And as part of that, I came to the United States for an exchange, and I met people at Caltech who were starting to think about biology simply as another field of application of control theory. And to me, that was mind-blowing. You know, all the math that I had learned that applies to springs and circuits and you know, water tanks, all that math I could apply directly to modeling a cell. So that was fascinating, and that's what led in the end to me reapplying for a graduate program in the United States, because I really wanted to learn molecular biology at a deeper level and understand how, like, the way we think about you know, self-driving cars, or can we get like reprogram a cell, but I really thought it's in the molecules. We need to understand how to engineer molecules to then prescribe how like a more complex organism will work. Yeah. So that's kind of like an overview of how this happened. So that how I ended up here.
No, because when I was looking at your biography, you had sort of 2 PhDs from very different places in quite different things as well. So I was intrigued to know what sort of like what path you took.
Yeah, a random walk.
That's great. I guess the next question kind of links to that a little bit, in terms of, so you can apply all this clever maths to biology, but what do you feel, I guess, are the biggest hurdles that have, because we still haven't got to the point where we can just imagine something we want to make with biology and we can go and create it. So what are the steps, do you think, in order to get to that point potentially?
Yeah, no, I think about that a lot. And in our, like, what I see is the promiscuity of biology is a problem, right? When you want to engineer pathways, you have an idea that you think of it as a circuit where things will interact in a very isolated way, and they will work beautifully. You characterize all the parts. You have this, you know, quality control over individual components. But then once you put everything together, because they're so promiscuous and there's side reactions, then everything falls apart. So sometimes I think, well, maybe it's because we're thinking as engineers who are used to working with wires and insulated parts. That's what the problem. Maybe because cells exploit this promiscuity, right? To introduce like the variability, the fluctuations, to actually get better at what they need to do over generations, of course, not over individual cells. But so maybe that we're simply thinking about biology in the wrong way. Sometimes I think we should really take a different perspective and just exploit fluctuations and variability and complexity, as opposed to trying to get rid of them.
Definitely.
But you know, you got like the different perspective of an engineer versus a biologist, I guess.
But I guess, like, so do you think then there's almost a hidden theory of engineering for biology? Do you think there are kind of similar laws or kind of, like you have, I don't know, Kirchhoff's laws for circuits, so there's similar things, but obviously written or working a very different way for biology?
Well, I don't know. I mean, in the end, the laws of physics and chemistry, they work the same way, whether it is a protein or a circuit, right? But then how we take advantage of the peculiar nature of proteins, that's the question. And I think, you know, the recent Nobel Prize to Francis Arnold, I think that's points into that direction, you know, directed evolution. Perhaps that's what we should use more, even when we build circuits for synthetic biology.
That's an important point, I think. I got one last question, which is kind of very, very general. So, so if you were starting out again, so if there was some student watching this, would you have sort of any advice for decisions you've made that you've maybe regretted later on, or kind of things that you wish you'd done that afterwards you hadn't in some sense?
I see. Well, maybe one thing that I repeatedly got wrong in my career was trying to work too much in isolation. And perhaps that's related to the fact that, you know, there's few women in engineering. And so it's maybe sometimes difficult to connect with your peers in a direct way. But I really wish I reached out more to my peers in developing projects, even just like chit-chatting, brainstorming. I didn't do that as much. So perhaps the advice would be, you know, like make more friends in your lab and outside of your lab, and just like because those informal interactions where you just, you know, grab coffee with someone and talk science, sometimes they give you really, you know, unexpected intuitions that really help you down the road. But and then, you know, you build your network of support. It's both productive and emotionally supportive for everyone. I wish I did more of that.
Yeah, I think it's quite poignant in today's world where things are becoming very isolated, and actually I think science needs to be very inclusive, otherwise you miss out on a lot of stuff.
Yes, exactly. Yeah, and we all feel it really badly now that we're all working from our desks.
Yeah,
but yeah,
it's really hard. Well, that's the last of the question, but I just want to thank you again for a really lovely talk, and I can't wait to see what materials you start making in the future.
Thank you. Hopefully we'll meet each other in person soon.
Definitely, yeah. No, I was thinking that it's frustrating with these seminars. Like, one of the nice things about giving a seminar is meeting people you probably wouldn't, and seeing what they're doing, and you wouldn't necessarily read the papers of that person, but then sometimes you find these weird connections and stuff. And yeah, I wasn't aware of all the things you were doing, and especially I work more in genetic circuits, but the material stuff I've got more and more interested in, because there's some real nice applications of some of this stuff, and the sorts of methodologies you're using are really they can connect really well with some of the transcriptional programs that we want to build in.
Yeah, no, I think so.
Right.
I think so. I think that if you use only transcription, you can probably adapt what we're doing very easily. If you can, I mean, and of course, I mean, it's likely that you will be able to control lots of proteins with RNA. So yeah.
That's cool. All right. Well, I won't take up any more of your time, but thank you so much for the talk.
Thank you for inviting me, and it was a pleasure to meet you. Thank you for giving me this opportunity.
Great. Okay.
All right. Bye.
Bye-bye.
Bye.